record because I didn't click record. But I guess I'll just try and find questions here that are actually useful to you. And we'll just go over those as your practice. Um, there's a couple questions in these, and then exam 3 has a lot that you guys can do as well, apparently. That I want to grab this. What are you looking at? Why are you looking at me for? Don't do that. Don't do that. I thought you said you didn't swing that way. You guys No, no, I'm not. It's staying in there. Anyways, um, okay, why don't we start with, no, there's not like a full test, so I'm just gonna go through in these, I'm just gonna use his study guide to find practice questions, because Holt gave us like questions to do, so I'm just gonna use those. So like for example, like 15 and 16, I think, or 15's right and 19. This is exam 3. This is this is exam 2 of 25. Is this one, or is it exam 3? Wait, what? No, I lied. This one. Exam 2 of 25 is what we're doing, but I want to look at this one. Where's 15? 15, 15, 15, 15. Is this the right one? 15 and 19? Yeah, I was gonna say, we did this before. This is the exam I did before. I'm not gonna go over that again. I'll do exam 3 though. Exam 3 for sure has quite a few that you guys can do. Yeah, exam 3 2020, this one. This one said that you guys can do question 8. Oh yeah, you can. Yeah, question 8. Yep. So you can do 8, 11. Yeah, let's start with 8 first. Um, yeah. So I guess what we can do this time is I can like go over them. Um, but I mean, maybe it's better if you guys take a look at it and then answer. But I don't know. Do people have a preference for how they want to do this? No. Okay. Whichever way you want. Okay. Cool. Um, I'll just like kind of go over my thought process for these then. So you wanna answer it? Okay, cool, yeah. What? Okay. So I will not pay attention to him. Mm. You had a chance to look at it? What are we saying the answers? The- I just asked you if you looked at it and you just said you did. Okay. For those of you who have looked at it, what is the answer? Okay, D, okay. Why are the others what? What are you saying? No, no. Well, you said it before anybody else even looked at it, so uh, the first time for sure. Yeah, so we can go over why the rest of them are wrong. So D is the correct answer. Why is this one wrong? Why is A wrong? It should be purines, right? This should be purine, not pyrimidine, because uric acid is a purine derivative, right? Why is B wrong? It makes uric acid, so it should be an upregulation of that, not deficiency. So it should not be deficiency, and it should not be pyrimidine here. Okay. Why is C wrong? What? Um, yeah. What does it regulate or what does it inhibit? Thymidylate synthase. Yes, it has nothing to do with it. Yeah. So I don't even care. Um, is this true? No, it's not. If it was soluble, then we wouldn't have gout. So this is not true. So E is wrong. Okay, good. Yeah. I feel like this is a pretty chill question. But you never know. So yeah, move on to 11. This one, I have been teaching you this since 440. I hope you can do this now. Single transamination reaction. Okay. Okay. Do we all know what the answer is here? We all said it's going to be glutamate, right? What which um which citric acid cycle intermediate turns into glutamate with one transamination? Alpha ketoglutarate. Thank you. Okay, which one turns into aspartate? Oxalate. Good. Which one turns? This is not a citric acid cycle one. Which one turns into pyruvate? Alanine. There you go. Yeah. Alanine. Yeah. Yeah. What about glutamine? Does glutamine have one? Which one is it then? In one transamination? I don't think so. You can do two though, right? So you can use from alpha ketoglutarate, you could transaminate and then add another ammonium group, right, with the glutamine synthetase technically, to make glutamine. That you could do. But for this question, the answer is going to be glutamate. The rest of these are not even like glutamine. We talked about you could make from that pathway, glutamine synthetase. Serine and arginine, these are like hand in hand if you do the THF pathway. So there's that. Arginine, you haven't covered. I mean, it's in the urea cycle, but you learned that yesterday, not today. Today's Tuesday. But yeah, okay, cool. Next we can look at 14. Um, okay, yeah. Which of the following accumulates in the absence of uh cobalamin? So B12. Why are you so clueless? Like all the time. Oh my goodness. It's a lifestyle. I wish I had like the slides that I could pull up for each of these individually. There's a lot of them though. Let me see if I can find the slide actually for that one, just so I can showcase that. Why would I pull up the video? Actually, you know what? Uh, you are right, but I don't want to. I should have my playlist bookmarked at this point, just to go back through it. I believe it was this one. Was it amino acid synthesis? It should have been. Where is it? This. Right. Should be this pathway. There. Yep. Right. So if you what? Yeah, yeah. But I would like look at this one basically. This is the main one. Um so for this one, hopefully you said it was the N5 methyl THF. That's the one that gets stuck there. That should make sense though, because the N5 methyl is the one that actually has the methyl group that ends up getting donated to the homocysteine to make methionine. And you need B12 for that reaction. So if you don't have the B12 in there, it just gets stuck as N5. and N5 just has a massive buildup in there. So hopefully that one makes sense there. So it should be this, right? In order to have a buildup of myth like any of these other ones, you'd have to mess with the other pathway. You'd have to mess with not this, but this, right? Because this is the other pathway that we talk about when it comes to these. So this is the other one that is important to remember. And unfortunately, I can't remember like any of these anymore. It's been a long time since I've done these. So, but hopefully you can see that these are just like it's most reduced, like middle, and then like most oxidized. Um, and so in terms of what actually gets receives the CH3 group, it's going to be the homocysteine to methionine. So that one receives the homocysteine, the CH3 group there. But yeah, very nice indeed. Uh, I think 14 through like 14 through 21. These are all questions you guys can do. So there's a lot of them. Okay. 14 through, okay, so we just did 14. Non-essential amino acids blank. So which of these is true for non-essential amino acids? B, okay. Anybody else got any input on that? Or are we just thinking B? You concur. Okay, cool. I would agree with B. So B is true. So non-essential ones are ones that we can synthesize, right? Which ones are essential? Like what does it mean for an amino acid to be essential? We can't synthesize it. So that would be like I think there's one. Yes, like C I guess would be like a maybe an example of a essential, right? If it was asked, huh? If if the question was asking which is true of essential amino acids, you would probably say something like C. So like, it'd be synthesized by plants and bacteria, but not by humans. So where do we get most of our essential amino acids from? Food. Okay, food and diet. Okay. Yep. So I agree with that. B is definitely the best answer. Have you guys? Oh, you have. Yes, you have. Yes, you have. Yep. Um. Okay. So what's expected for 16? Yeah. This one's kind of hard. 16 is kind of tough. But what are we thinking? Also, read the question, please. Not be expected. Okay. Anybody else have any input? Just send her. Yeah. Anybody else got input? So which of them would not be expected? Would you expect to be there to be impaired beta oxidation? You would. Okay, why? Okay, and then why is that important? Right. Okay, so the carnitine, right, is used to bring the fatty acid chains into the mitochondria to do beta oxidation. Okay, so it should be impaired, so that's true. Um, would the fatty acyl-CoAs accumulate in the mitochondrial matrix? They wouldn't. Okay, so that I think that's a good answer, right? Um, would still be able to mobilize stored fatty acids from adipose. Um, and serum levels of fatty acids would increase. Yeah. Yeah, right. Because it's going to seem like we don't have a lot of energy, right? So maybe it would start mobilizing fatty acids, right? So that we could try and produce more energy, right? So this could be possible, right? So that's reasonable. Um, right? This makes sense as well, right? Because if you're doing less beta oxidation, you're producing less acetyl-CoA. You can't make any ketone bodies if you don't have any of that. And then E is just that. So I would say B is the best answer here. Does that make sense to folks? So yeah, while we're doing that, okay, cool. Okay, 17. Which of the following statements describes a general feature of the lipid bilayer? Okay, this one I think is fine. So in terms of the lipid bilayer, all right, yeah, I guess we can just go over them one by one here. So I'm actually gonna start from let's start from D. So is the bilayer stabilized by covalent bonds? No, okay. I sure hope not. Um, is this true? Polar but uncharged compounds readily cross the lipid bilayer. No, okay. How do you cross the lipid bilayer? What's the best way to do so? Like what characteristics would you need to cross the bilayer? Hydrophobic and ideally small, right? Maybe small polar things can cross, like water sometimes crosses, but not very often, which is why you need aquaporins. But generally speaking, it needs to be small and hydrophobic. Okay. Individual lipid molecules in one leaflet undergo transverse diffusion to the other leaflet of the bilayer. Yeah, right. So I mean, this is generally not the best in terms of like it usually doesn't happen. Usually you need ATP for something like this, like flipase and flopase, right? Those usually require those. Um, but yeah, individual lipid molecules are free to diffuse laterally in the surface of the bilayer. Is that true or not? Okay. What kind of technique would you use to test the diffusion of yeah, frap, right? What is frap? Like how do you do frap? Like what does that entail? Well, yeah, okay, well you have to do something before you bleach it, but you add fluorescence to what? Okay, to all the lipids, right? And then you what? Then you photo bleach it, okay? And then after you photo bleach it, what do you do? Yeah, okay. So how do you know, like, how do you see the diffusion rate though? How is he on top of it more than you guys are? What's going on here? Yeah, yeah, yeah. See, it's like, yeah, yeah. So it's like once you photo bleach it, right? There's like a white spot, right? And then over time, the fluorescence diffuses inward, right? And it starts to get rid of that white spot, right? That's how you know that these things are able to freely diffuse across the membrane. They don't have to worry about any of that nonsense, right? Okay, good. Yeah. Alrighty. For 18, which of the following within the inner core of the liquid, which of the following statements correctly describes molecules that are within the inner core and those that are located on the outer shell of lipo? Okay, yeah. Yeah, just be careful with reading this one. Huh? Yeah, this is kind of a bit of memory. I mean, you can probably you can work through it. No, you can work your way through it. Huh? Okay, so let's look at A, right? So A, do I expect to see phospholipids in the core of my apolipoproteins? Why not? Because they've got polar head groups. Okay, yeah, I shouldn't expect to see that. What about free fatty acids and triacylglycerols? I should expect to see those inside because those are all free fatty acids is a bit weird because free fatty acids they may have a negative charge. So maybe not. I'd expect maybe more there to be like more of these the triacylglycerols and diacylglycerols. That's probably more reasonable, but definitely the phospholipids should not be in there. So A is for sure out of there. The inner core contains apolipoproteins, triacylglycerols, and free cholesterol. Right? Do I expect to see apolipoproteins in my core? I sure hope not. Right? There's apolipoproteins are on the outside, right? They help with docking. So when you dock to a cell to allow for absorption of the fats and the cholesterols, um, you use the apolipoproteins, so they're definitely not on the inside. We already talked about how the inner core should not contain phospholipids. So that one's out of there. So the inner core contains triacylglycerols and cholesterol esters, and outer shell contains phospholipids and apolipoproteins. That sounds better, right? So D is okay. Why is it that the inner core has cholesterol esters and not just normal cholesterol? It's more hydrophobic, right? So it leads to better packing. So when we are able to better pack, we can store more and then we can deliver more. But yeah, other than that, good, good, good. And then for 19, the inner core contains cholesterol. Contains. Uh, which of the following domains of the fatty acid synthase catalyzes the reaction shown below? Okay, cool. Yeah. Okay. So what kind of reaction is this? So we're removing a water, right? And then we're forming a double bond, right? That's the big thing here. So it's going to be some sort of dehydrogenase, right? And is it this a reduction reaction or an oxidation reaction? Is it an oxidation? Right. So you're basically adding or oh sorry, hold on a minute. No, 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 no. Wait. Um actually based off these answers, huh? I was gonna say based off these answers you actually wouldn't, right? So what is the you could maybe consider it an oxidation or reduction, but there's actually a better answer in these choices. Right. So dehydration, right? So you expect some sort of like water removing thing, right? And so which one of these removes water? The one that hopefully says dehydratase, right? The one that hopefully says get rid of water. Um, so yeah, I think I said that you don't really have to memorize the names. I think you should just like I think the best thing to do is like look at the ends. This is usually enough to get by. But I mean, it doesn't hurt to know what the names are. So by all means, go for it. Which of the following statements about the synthesis of cholesterol is not correct? Okay, let's see here. Okay, yeah, so this is a good one. All right, yeah, cool. All right, let's start with the top one. Is A true? Is it? It should be true, right? So all carbons of cholesterol are derived from acetyl-CoA, right? All of them. Right, which is why you don't make cholesterol under starved conditions. Right? You don't make cholesterol under starved conditions. It's also why glucagon inhibits HMG-CoA reductase, right? So you don't have that active. So this is wrong. This is true, right? It would actually occur like that. Um, is this true? Which enzyme is that true of? It's a synthase, right? I'm pretty sure it's a synthase. Fatty acid synthase is one pain, right? But it's not true of acetyl-CoA carboxylase, maybe. Might have to look that up afterwards. But it's definitely not HMG-CoA reductase, right? Because that's the main one I think that they're talking about here. But also like all the enzymes are not single glypeptide chains, so cool. Wait, sorry, that's the right one. But let's talk about the other ones anyways. So HMG-CoA reductase catalyzes the committed step in cholesterol biosynthesis. That is true, right? That's like the main one that you guys are like expected to know. Everything else for the cholesterol pathway, I honestly don't care. But that one you should know. You should know for sure that that one's the one that's regulated. It's upregulated by insulin, downregulated by glucagon, all that stuff. Cyclization of squalene generates the fused ring structure. Yes. Squalene is like the weird Squalene is this guy. It's like all the weird ring. Yes, like see this thing? So squalene, right? And then when it fuses into the cyclized version, and then you go from there and you can make your cholesterol, right? So, yep. I think the last one that's and then for 21. Okay, yeah. So which of the following situations will result in an increase in ketone body synthesis? So when do we start making ketone bodies? Fasting, right? And so that means we've run out of what? So you've run out of glucose and you've run out of fatty acids and you've run out of like pretty much everything, right? So ketone bodies are like your last resort. Can you use proteins to get ketone bodies? You can, right? So I think you guys yeah, that's like in the last lecture you guys did where it's like ketogenic and glucogenic amino acids, right? And you guys haven't, it's not on this exam, it's on the final, but like leucine, lysine are ketogenic amino acids. Those turn into acetyl-CoA, which can be used to make ketone bodies, right? So you can use proteins to do this. So if you run out of like everything, right, you will start degrading proteins and using them to make form energy. So yeah, that's one way of looking at it. So yeah, so which one would result in an increase in ketone body synthesis? So would having high insulin cause ketone body synthesis? No. Okay, and it wouldn't cause it because of what? Why? What does high insulin signify usually? Usually you have a lot of sugar, right? You do all that stuff, right? Yeah, yes, that's another way. Yeah, that's a good way of thinking about it. Beta oxidation's inhibited, right? So you can't use the acetyl-CoAs to make ketones. Um, High citrate levels in the cytosol. What does that do usually? How is citrate used here? What? Yes. Okay. So, what part does citrate play in that? How does citrate feed into the fatty acid stuff? Citrate lyase, right, to make acetyl-CoA, right? But I mean, like that wouldn't really increase ketone body synthesis, right? Because like we're putting acetyl-CoA out there, but I think the main one, I actually can't remember the glycerol part. Glycerol 3 phosphate. Um, I'm pretty sure, yeah. They'll just make more of them. But I think the main thing to keep in mind here is this, obviously, um, doesn't really work. If you have low levels of it, you can't really make ketone bodies because you need acetyl-CoA to make ketone bodies. But if you have low levels of oxaloacetate, then all of the acetyl-CoA that you make just builds up. And then over time, if you don't have oxaloacetate to attach the acetyl-CoA to, you'll just use the acetyl-CoA to make ketone bodies over and over again instead. So if you have low oxaloacetate, right, and you have high acetyl-CoA, in order to make citrate to go through CAC, right, you need to combine a oxaloacetate and an acetyl-CoA. If you don't have oxaloacetate, the acetyl-CoA is just there. It's not doing anything. And so in order to use up the acetyl-CoA, you'll start making ketone bonds because that pulls all the reactions down, right? So if you run out of oxaloacetate, you start doing ketone body stuff. Yeah? Low levels of acetyl-CoA, like I said before, doesn't really make sense here because you use them to make ketone bodies in the first place. So that's why I would say the E is probably not a good answer there. And I don't remember exactly. I can't remember the reasoning behind C. I'm not gonna lie. Um, yeah, I can't remember the exact reasoning behind C, and I also a little bit fuzzy on B. The citrate levels of cytosol. Can't remember that too well, but either way, D is for sure the best answer here. And let's see which one. What else can we do in this exam? I'm assuming that's what they're getting at here. I think if there's high levels of this, I think you're right. It would just lead to more. fatty acid synthesis, but I can't quite remember off the top of my head. So, but we can do 32 to 42. So yeah, okay, you have to, these are a bit, oh, okay, yeah, all right, yeah, let's do these then. These are always fun, aren't they? Okay, 32. So starting with this one, in aminotransferase catalyzed reactions, glutamate plus wait, have you guys done this? Yes, you have. Yes, you have. Yeah, you can. Yeah. What are the products here for this? Okay. And alanine. Okay, good. So yeah, it'd be alpha ketoglutarate. I'm just gonna call it 8 kg because I'm lazy. And alanine. Right? So yeah, alpha ketoglutarate and alanine. All you're gonna do is take the N off of glutamate and um switch it for the carbonyl group, right? So yep, good. Uh what cofactor do all transaminases use? PLP, good. Okay. What cofactor do decarboxylations use? Decarboxylations? Carboxylases use biotin. What's the one that decarboxylases use? T. TPP. Yeah, yeah. Oh god. Well, you have to know it for the MCAT, so you better know it. um. Oh, I guess you guys haven't done this. Um. I'll just. It's in the lectures, but you guys don't have to know it. It's leucine and lysine. Why did he put that in the? Okay. Cool. Whatever. It's fine. Um. 35. Oh yeah, yep. You guys have not done this. Yeah, it should be glutamine and alanine. Yeah, but you guys haven't done this yet. Okay, cool. Yep. Get rid of that. Nope. You haven't done 36 either. Well, no, you have done it. It's just you're not using it. Oh, okay. How convenient. What reaction is this? Okay. What's the cofactor? Biotin. Yeah, it's a carboxylation reaction. If you ever see carboxylation reactions, usually biotin. So, yep. What number is biotin in terms of the vitamins? B7. Okay, good. Yeah. What's PLP? 6. B9 is folate. Yeah, yeah. I'm gonna have to like bleep the video out with how many times you've been swearing, dude. Anyways, okay, yeah, I think you were just asking me about this. What enzyme does this? I guess it's a bit different, yeah. This is honestly, I'm gonna be honest with you, this is one of those questions where you might just need to put in your note card. There's literally like no way you're memorizing this. So, I mean, unless you're like crazy. This is a bit of an annoying one, I'm gonna be honest with you. I'm pretty sure it's in one of the slides. Because there's like phospholipase C and there's like the other ones as well. Oh, did you only use those two? Maybe not then. Okay, if that's the case, then maybe not. This one should be phospholipase D, but if you guys haven't done, okay, cool. Then I wouldn't worry about it then. Okay. Oh, did it? Oh, did it say you can just say phospholipase? Okay. Yeah, I mean that. Yeah, I mean if they let you just put phospholipase here, I would probably give you credit for that. I mean that seems reasonable because you don't need to know the whole actual thing. So, okay. When energy in the oh yeah, this is a good one. Where what protein or thing or molecule are these things carried on? Serum albumin. Yes, yep. So albumin, right? Albumin's the blood carrier protein, remember? And why can't I just put fatty acids in my blood? Because they aggregate and then they'd be bad. right? Because they're mostly hydrophobic. There is some hydrophilic side to them on the heads, but if you especially if you're transporting triglycerols, those are not hydrophilic at all. So they would just like become soluble, insoluble plaques if you just let them sit there. No, no, that doesn't mean anything. Serum is just a liquid. So no, you gotta say albumin. Yeah. Yeah, if you just you can say serum albumin, you can say albumin, whatever you want. You just can't say serum. Um, Once inside the cell requiring the fatty acids are carried to the mitochondria using what do they use here? As the how do they get into the mitochondria? Carnitine. Yes, the carnitine shuttle is the main way of of grabbing and pulling pulling stuff into the cells, right? Right. Okay. Uh, I think we can do yeah. I think we can do all three of these. Yeah. So reduction of uh what molecule? Um which one? DHAP, dihydroxyacetone phosphate. Good. Yep, dihydroxyacetone phosphate is the one that turns into glycerol 3 phosphate. Good. This one is a bit mean. There is a slide on it, and I do remember doing it actually. Hormone sensitive lipase. Which version of the fatty acid does it, or the glycerol does it actually hydrolyze? Diacylglycerol. Yep, the diacylglycerol. The triacylglycerol and the monoacylglycerol are from different things. Yeah, yeah. That's a mean one. I wish I'd have to look at it again, but I can't remember off the top of my head. So it's just kind of there, right? Yeah. I think it's just like because most of the time you don't have a lot of monoacyl glycerols, it's generally just typically active all the time. And because in order to get to a monoacyl glycerol, you have to get rid of a triacyl and then get rid of a diacyl. So, and also the triacyls and diacyls don't fit in the active site of a monoacyl glycerol unless you cleave it properly. So, I think for that reason, you can just say, oh, it's generally just constitutively active to some extent. So, but yeah, either way, even if you are not expected to know the mechanism of how it's turned on, if it's not in the slides, then it wasn't in the slides. So, I wouldn't worry too much about that. Uh, let's see. Right, so the synthesis of what is the rate limiting step for cholesterol synthesis? I actually got this question wrong when I took this exam. No, it's not HMG-CoA. Yes. HMG-CoA reductase reduces HMG-CoA to mevalonate. Right, so it's meval. I like how he's nonchalant with it. As long as he's so nonchalant, he just knows. Yeah, so how do you spell mevalonate? I can't spell it properly. Mevalonate. There you go. Yeah, I got this wrong. I put HMG CoA and it's not HMG CoA because it's asking about the product that you make from it, right? So HMG CoA reductase makes mevalonate from HMG CoA. True, true. See, reading is important. Um, yeah. I think is that it for this exam, or is there more? 32 to 42. 45? Okay, yeah. We can do 40. 45 is on your exam, so. Oh, did he say 23 isn't? Bro. Dang, you guys are just. Oh, hold. Is it on the study guide? Let me look at the study guide. Is it through 23? Someone double check that, please. He's cooked, chat. On the ninth, for example, study guide, key terms and preparations only go up till 22. It's on like if you look through the study guide to the end, it won't end. Doesn't say 22. Okay. Okay. In that case, I'll just go past these then. Yeah, okay. In that case, I don't think you guys need to do 45. And then 46, I think for this one you can do. So for A, you can do. So what enzyme converts glutamine to glutamate to glutamine? Synthetase. And what do you need? What are the reactants required? What? And ammonia. Thank you. Yeah. So it's gonna be glutamine synthetase. I'm not gonna write it out 'cause I'm lazy. But you also need ammonia and you need ATP. So that's good. And then for B, mm, this is a bit of a. I mean, yeah. I guess you guys could. I think you can maybe infer through this, right? So glutamate to glutamine gets rid of an ammonia, right? Is ammonia good or bad for the body? Bad. Okay. So um the metabolic conversion of glutamate to glutamine accomplishes basically just the removal of toxic waste, right? So it removes ammonia, and therefore your brain doesn't explode, right? So that's the main thing there. Um, this I don't think you need to know. This is not. This is for the final, but the glutamine is then shuttled to the liver, right? And then is put into the urea cycle. Then you can secrete the urea, right? But again, I don't think this is important for this exam. And then the amount of glutamine produced within the brain tissue can exceed the amount of glutamate taken up by. This is a fun one, actually. The amount of glutamate taken. Right. Okay. Yeah. So how would I end up making more glutamine than normal if I'm taking up glutamate and then adding amines to the ammonia to it to make glutamine? How else can I make how else can I make glutamine? Alpha ketoglutarate to glutamate, and then glutamate to glutamine. And that's another way. So that's what they're trying to get at here. So what mechanism is responsible for the production of the additional glutamine? It's going to be that, right? That process of going from alpha ketoglutarate to glutamate. What enzyme does alpha ketoglutarate to glutamate? Glutamate dehydrogenase. Right, good. Okay. And then yeah, I believe it is in urea cycle, yeah. Yeah, you guys haven't learned urea cycle. Wait a minute. Did you guys not do urea cycle yesterday? Was that on Monday? Yeah, yeah. Okay, it's not on the exam, but it was yesterday, right? I thought it was yesterday. I looked at the lecture yesterday. I thought it was urea cycle. Oh, okay. Yeah, yeah. I'm pretty sure it was yesterday. Because I looked at the slides today. Because I was wondering when you guys were gonna do urea cycle. Um, yeah, the orders all whack out because you guys have done urea cycle last. We did it like a couple weeks beforehand. So I'm like trying to figure out what you guys are doing. No, the urea cycle is the last one, I think. This is the only one they put on the canvas. So I'm gonna make the assumption it's the last one. Yeah, I hope it's the last one. I need this guy is so done. No, you're all good. You're all good. Don't worry. I'm feeling the effects of it as well. But yeah, I think that's pretty much. Does anybody else have questions about like content questions? Because I can just keep going through questions for like the next 10 minutes. But if people have like specific content questions, Yeah, I trust you. I trust the study guide more than what? What? Like, yeah. Don't think that's that important. Yeah, I mean, if he tests you on that, I feel like that's kind of insane. Yeah. Uh. Yeah, yeah. I don't know. I feel like that's a bit out there. I'm not gonna lie. Like that'd be kind of insane. I mean, so every now and then they do that weird stuff like the AAU question on the on the last exam. That one's that one's fair. I mean, I feel like there's like one or two questions, but that's just how Goo tests. I feel like Holt is a little bit more reasonable with his exams. This one, third question. This one, yeah. This is like a Dana question. Probably not. No, I don't think this is that important for the version of biochem right now. So I wouldn't be too concerned about it. Um, yeah, that's some Dana stuff right there. That's Dana Miller right there. I'm pretty sure it's Dana Miller. So, let's see here. I think are there a couple more? Like exam three, supposedly this one has like 1 through 8. Oh yes, finally a freaking fatty acid question. Which of the following statements is false and um, okay, yeah, false. Okay. So for this guy, I didn't mean to do that. Whoopsie. So for did I have to trying to pull up the key so I don't lie to you guys. Okay, so I think we can do 1 through 8 is what it said to do. Yeah, let's just do as many as we want to, guys, I guess. Okay. Which of these is false? So, oxidation of 1 gram fatty acid yields more energy than oxidation of 1 gram of glucose. Is that true or false? True, right? Yeah, it's like I can't remember the exact like number. I think it's like 37 kilojoules per fatty acid, and like I don't know, like 16 or something per. Yeah, yeah, fair enough. Yeah, me too. Yeah. You just need to know it uses more. Triacylglycerols must be highly hydrated when stored in organelles known as lipid droplets. Is that true? Low key, I sure hope it's not true. Low key, you don't want putting water in my oil right now. Yeah, no. So that's definitely not true. So B is for sure false, but we can talk about the other one. C, um, triacylglycerols must be digested before they can be transported into. Yep, okay, that's true, right? They need to be digested. You can't just put a triacylglycerol straight into the cell. It's too large. In circulation, free fatty acids are, but yep, this is true. It's not just bound to serum. I'm just saying, it's bound to albumin. Okay. In the liver and epithelial, triacylglycerols are assembled into lipoprotein complexes, right? This is true. How do they get from the stomach into the liver? What are they sent in? Chylomicrons, right? And then where do chylomicrons get? So excited about that. What system do chylomicrons enter first? Blood circulation or lymphatic system? Lymphatic, yeah. They enter lymphatic system first, and then the blood circulation. Okay. Yep. Okay. Um, which one of the following fatty acids can be made from 16,9 in humans? Yeah. So I guess the question is like, what are the desaturases that humans have, first of all? 4, 5, 6, and 9. And when you add carbons to these fatty acid chains, where do you add them? At the end or at the start? Start, right? So basically, if I have let's say I have a double bond at carbon 4, right? And I add a 2 carbon chain, that double bond now moves up to 6 because I've just added 2 carbons before it. So you would have to go through individually and just do each one basically. So like for this, um, I'm always forgetting 4, 5, 6, and 9, right? Is the thingies. So for you'd have to like go through and say, okay, how many carbons am I adding in this first answer? I'm adding 8. So if I add 8 carbons, this 9 will move up to a 17, right? And so you're basically just adding double bonds in every couple. So like for this, I'm trying to see if you add 4, 5, 6, and nine. Where is it? I'm trying to see how you can add this to 15 because you need to add one so it becomes 15. Mm. But I mean, yeah, basically the idea is that you can add, basically if you add the double bond and then you extend the chain, you'll make it so that all of these are actually possible. And so the answer would be all of them for that. Just because I'm trying to remember how you get the 15, but I can't remember. Yeah, no worries. I was just trying to remember how you'd get the 15 one, because the 15 one is a bit strange. 4, 5, 6, and 9. I mean, you would make the chain first and then desaturate afterwards if you wanted to desaturate it. There is just specific enzymes that do it. I think it's 4, 5, 6, and 9. I'm pretty sure. I can't remember is it 4, 5, 6, and 9? I think it's 4, 5, 6, and 9. I'm like doubting myself now. So I think plants have 11 and something else. So 12 and 15, yeah. It's 5, 6, and 9. Sorry, not 4, 5, 6, and 9. No, no, 4 is there as well. Yep, 4, 5, 6, and 9. Yep. And then plants have the extra ones, which is like I think you said 11 and 13. Or 12 and 13. 12 and 15. Yeah, these are things I put on my note card when I was studying for this, I'm not gonna lie. Yeah, definitely 12. Yeah. So for that one there, that's what I was trying to figure out here exactly. If you desaturate it at 9. Yeah, 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 yeah. Yep, that's true. Yeah, I think that's the way to go. Yes, yeah, yep, that's the way to go. Yeah, you would add 2, that would get pushed to 11, that one would become 9. Yeah, there you go. Yep, and then you would just extend all the way to 24, and then you would just do 4 and 5, and you'd be good. Yeah, and the same thing works for the rest of these as well. Yeah, these are all a pain. Yeah, these are the most annoying questions for sure. They take so long to figure out. So, okay. Um, yeah. I mean, got what, like 2 minutes? I guess I'll ask a couple more. Uh, 4. What's 4? Which of these can be converted directly? Okay. Yep. What is this? What's this guy? Huh? What amino acid is this? Glutamine. What's this? Aspartate. What can this be converted into? Acetate and asparagine, right? Yeah, yeah, yeah. Huh? He's gotta remove the. Remember, there's a synthetic. Yeah. Okay. What the enzyme that does this? What does it use as the nitrogen donor? This was in your slides. I remember this. Glutamine. It uses glutamine as the donor. Yeah. So yeah. And then this is asparagine. And what is this? I sure hope you know what this is. What? Is it spicy? Yeah, so what is this? It's alanine, right? And then what happens if I remove this N, replace it with a carbonyl? Pyruvate. Yep, it's pyruvate. Yep, good. Okay. All right, we're done.